Hello everybody. My goal today is to defend the LDL, especially the oxidized LDL, and try to convince you that the oxidized LDL is an injured fireman and not an arsonist. And you'll understand what I mean by that. And by the way, in the background is my bicycle. I've been coming to work for the last 6 weeks on my bike, not used the car, having an absolute blast. Mainstream medicine has the current paradigm that LDL is uniformly a bad player, that it has no real biologic functional role that is beneficial. And they view LDL as something that gets into the blood vessel wall. Underneath the lining of the blood vessels, which is the endothelium, is a layer to which apparently the LDL is getting attracted, especially oxidized LDL. And oxidized LDL is damaged LDL, LDL that has gone through oxidative or burn injury. And the oxidized LDL initiates an inflammatory cascade, inflammation that attracts macrophages. The macrophages then ingest the LDL and the macrophages then become a fatty streak or fatty plaque, and that is the underlying cause of blockages in the blood vessels that leads to heart attacks and heart disease. My three heroes are shown on this slide. That's Dr. Ravneskov, Dr. Kendrick, and David Diamond. And these people have done work that contradicts some of the traditional view about LDL, because they have demonstrated adequately that in population-based studies, when you take large number of patients, after age 50, higher the LDL, the lower the mortality. The higher the LDL, the lower the risks of having infections, the lower the risks of having malignancies, the better the memory. So their paradigm of high LDL, and by the way, high LDL and cholesterol go hand in hand. When you have a higher LDL, you'll also have a higher cholesterol, and vice versa. So my heroes have been talking about a different paradigm, and my clinical experience, I've been a cardiologist for about 30 years, and have observed that in a number of my patients who are in their 80s and 90s, that they have robust health when their cholesterol levels are over 300, when their LDL levels are over 200, and in many of these patients, I have done a coronary angiogram for clinical reasons, and I'm letting that play out there, and the coronary angiogram is showing the right coronary artery out here, and it really does not demonstrate any blockages. So these are 80 and 90-year-olds that are defying the conventional wisdom, in whom they have had high cholesterol all their life, and yet they have not had any significant blockages of the blood vessels of the heart. So I think as a low-carb practitioner, I have viewed cholesterol not from a standpoint of whether it's high or low, but from the standpoint of cholesterol quality. And I have said that many times before. Good quality cholesterol is one in which the triglycerides, or the fat in the blood, is low, and HDL, which clears fat from the bloodstream, is high. That these people are insulin sensitive, in other words, they are not pre-diabetic or insulin resistant, that they have low inflammation markers, that their adipose tissue or fat cells are healthy. And you can find that through different methods. One of them is adiponectin, that their muscle mass is high and that their fat mass is low. So many of these patients show these clinical features. These are their characteristics. That is, insulin sensitive people who have high cholesterol or high LDL. They're physically active, they have normal body weight, they have high muscle mass, or the percentage of muscle mass compared to their fat mass is high. That the high LDL perhaps gives them the opportunity to resist viral infections, bacterial infections, and that's especially relevant in this current pandemic, that they overall have a sense of well-being based on my clinical experience on evaluating a number of these patients for the last 6 years, and that they overall have a high energy level. This got me thinking into exploring a little bit more about the oxidized LDL. What is LDL oxidation, and does the LDL have mechanisms to prevent oxidation? And yes, it does. The LDL has a number of molecules inside it. Vitamin E, which is called alpha tocopherol, ubiquinol or CoQ10, beta carotene, gamma tocopherol, alpha carotene, lycopene, and many others. Now, out here is shown an LDL molecule, and I have gone through that a few times before. The LDL molecule, in order to dissolve in blood, has an outer cover of phospholipid that makes it water soluble. It's got an identifying protein. In this case, it is the ApoB100. And the cargo of this LDL molecule is not just triglycerides and cholesterol, but it has these antioxidants. And in here I have taken the opportunity to show you 3 important antioxidants, and that's alpha tocopherol, the CoQ10, and the beta carotene. The alpha tocopherol is perhaps present in the highest quantity. And the way our body functions is that the LDL protects itself by removing oxidative damage or helping counteract oxidative damage through these antioxidants, principally the vitamin E, the CoQ10, and beta carotene. So these are used first before the LDL molecule itself gets oxidized. And here is a paper, and I have put this paper out to show you that if you increase the amount of vitamin E in our LDL, then vitamin E protects the LDL from oxidation. So this is all that this paper is trying to tell you, that they have done studies in which they have shown that when exposed to burn injury or oxidative injury, if you load up the LDL particle with vitamin E, the oxidation of LDL itself is protected. And why is this important and why is it relevant? The reason it is because some of the medicines that we use, and I'll be discussing PCSK9 inhibitors, reduce the levels of vitamin E in the LDL. So here is an LDL molecule, and I have shown the LDL molecule as being oxidized. It's darkened, unlike this healthy LDL molecule. And this oxidation of the phospholipid bilayer and of the apolipoprotein happens only after it has used up the vitamin E, the CoQ10, and the beta carotene, at least according to some of these studies. So when viewed from this standpoint, the LDL is actually a defender of oxidation. It is something to be cultivated to protect us from oxidative injury. It's not causing the oxidation, but it is soaking it out. This is a study on PCSK9 inhibitors. This one is with Repatha, I think. And what PCSK9 inhibitors do is that they upregulate the receptor in the liver that takes away the LDL from circulation. So when you upregulate the receptor, you reduce the amount of LDL. The LDL levels fall dramatically low to about 30 milligrams per deciliter. And this makes physicians happy because they can demonstrate that they have removed and reduced a molecule that has been known to cause heart disease, at least in their paradigm. The patient feels good because the doctor is happy and he can see an actual change in a blood test number. But what physicians don't know, and if you were to go and ask maybe 100 cardiologists whether use of PCSK9 inhibitors reduces vitamin E and is that of any relevance, I do not think that many of them would actually be giving you an answer. But in this study, just 52 weeks, about a year, the reduction in vitamin E in serum was 26%, as shown in the left part of the slide. And when you looked at the reduction in vitamin E in the LDL molecule itself, it was a whopping 61%. And our studies that I pointed out to you before are showing that the vitamin E is an important antioxidant component of the LDL molecule. On the right side is the control group, and the control group did not have any significant reduction in vitamin E either in the serum or in the LDL. Now in this study, the slide that I just showed you was a subgroup, 100 patient subgroup that was studied extensively. But this is the larger overall population. I think there were over 1,000 patients in this study. You can see on the left side that in 52 weeks, compared to the placebo group, which is pointed out here with this arrow, the drug-treated group had a significant reduction in total vitamin E. But the reason I felt compelled to include the slide is out here is showing different groups depending on the level of reduction of LDL. The greater the reduction of LDL, the greater was the reduction in vitamin E levels. I think that this is data that we should strongly look at before we decide to use a medication. You know, I'm trying to do these talks on my own and I forgot to turn on the light. So now I'm going to turn on the light and see if the recording is better. Moving on, let's leave oxidative damage in a little different direction and focus on how the LDL molecule works to help fight against bacterial, viral, and fungal infections. So in this slide, it is showing that the LDL molecule does many things. One of them is called Quorum Sensing. We're going to get into each one of these individually. It neutralizes products of gram-negative and gram-positive bacteria. These are different kinds of bacteria that either make the LPS or the lipotoic acid or LTA. There is also binding of the viral particle by the LDL. It's called lipoviroparticles, and I will define that a little bit further. So what is quorum sensing? So in this slide are shown bacteria. These are Staphylococcus aureus. And what these bacteria do when they get into our lungs before they set pneumonia is to release a protein. This is called the AGR protein or accessory gene regulator protein. And the function of this protein is to go and check out the milieu, the surroundings. So it's sending out a signal to see if the environment is favorable to multiply. So the bacteria are sensing, hey, is this good area where we can multiply and cause an infection? So this AGR protein comes back and tells the bacteria, yeah, the environment is favorable, let's multiply. So what happens in this situation is that as this thing comes in and tells us the environment is favorable, the bacteria start multiplying, they gain a foothold and cause an infection. Now this is called quorum sensing established bacterial multiplication. What is the defense in our body to prevent this bacterial multiplication is the LDL molecule. The LDL molecule comes in, it soaks up the AGR protein, it prevents quorum sensing, and it neutralizes bacterial virulence, or the chances of the bacteria causing infection, and it reduces the risks of us getting infected by the Staphylococcus aureus. So this is one of the functions of LDL. We also mentioned that these gram-negative and gram-positive bacteria, different type of bacteria, produce LPS, lipopolysaccharide, and lipoic acid, and these are endotoxins. These are bacterial products that cause inflammation. I have borrowed this cartoon from Gary Fettke. This is his way of depicting inflammation or burn injury. Burn injury leads to cellular death or necrosis. What mechanisms do we have to neutralize the LPS and the LDA? The mechanisms that we have again is the LDL molecule, the apolipoprotein B100 and the LDL molecule come in and bind to LPS and LTA and reduce the risks of inflammation and hence protect the body and hence prevent us from having bacterial infections. So this leads me into the question that there are people with high cholesterol levels, familial hypercholesterolemia, who have high LDL levels. What is the mechanism of that? So this is a slide that is showing LDL molecules out here, and the regular cycling of LDL molecules happens through these LDL receptors that I'm pointing out to here. The LDL molecules bind to the LDL receptor, they're taken up by the liver, and the liver then subsequently recycles the cholesterol, recycles the LDL receptor, because it does not, the body is very nicely designed, it does not want to waste material. The familial hypercholesterolemia people are people who have defective or absent LDL receptors. So since the LDL receptor on the liver is not functioning well, the amount of LDL or bad cholesterol in the bloodstream is very high. So one would expect, if our theory or our hypothesis that LDL protects against infection is right, then we can show in animal models of an LDL receptor knockout mice. So these are mice in which the LDL receptor is absent, their LDL levels are high. So you would expect that in these mice, that they would be protected if they were injected with lipopolysaccharide or if they were injected with bacteria itself. So out here is LDL receptor deficient mice that have a high LDL cholesterol, 9.55 millimoles in this rat, compared to a wild type litter mate in whom the LDL receptors are normal. His LDL cholesterol is fairly in the normal range, 2.25 millimoles, European units. So both these groups were exposed to either lipopolysaccharide, which is bacterial product, or the bacteria itself. In this case, it's called Klebsiella pneumonia. What happened? So in this slide, it is showing that the normal wild type mice with normal cholesterol, when given the LPS, the bacterial product, they died very rapidly. 100% of them were dead depending on the amount of the LPS that was injected. What happened to the receptor deficient mice who had high LDL cholesterol. They required a lot higher bacterial product before inflammation set in and killed them. The reason for this is that the high LDL was soaking up the lipopolysaccharide and preventing inflammation that caused death in these animals. And that is demonstrated in this slide. When you measured the amount of inflammatory mediators, and this is TNF and interleukin alpha, these are markers of inflammation. What was found is that the inflammation was relatively lower in the mice that had high cholesterol as a result of receptor deficiency compared to their wild type mice. In the process of neutralizing the LPS and in the process of dealing with bacterial infection, you can see that cholesterol is being used because this is the cholesterol level before infection. This is at 90 minutes after the infection or after administration of LPS, and you can see a substantial amount of cholesterol has been used both in the receptor deficient mice as well as in the wild type littermates. But one can see that the wild type littermates had much lower cholesterol to begin with, so they could not have the amount needed to neutralize the bacteria and bacterial products compared to the mice that had LDL receptor deficiency in whom the LDL was much higher. Now there is a recovery of cholesterol as the mice were getting over the infection. As you can see, at 24 hours, there was some improvement in the amount of cholesterol present both in the receptor deficient mice as well as in the wild type mice. Now this slide is trying to tell you what happened when actual bacteria were injected into these mice. So here is the wild type mice that is getting the Klebsiella pneumonia injection, that is bacterial injection. All of these mice, there was 100% mortality by about a couple of days. Whereas in the mice that had LDL receptor deficiency and high LDL, you can see that although a substantial number of them died, it took them some time to die, but about 30% of them remained alive, indicating that high LDL in these mice was protective. Now, I don't want to get into the detail whether we have human studies. I have shown that before to you. But I again wanted to give credit to my three heroes because they have done adequate work that shows that humans with familial hypercholesterolemia who have low amount of LDL receptor, who have high LDL levels, are clinically protected from infections in large demographic studies. But I do want to use this particular study to explain and illustrate an important point. And in this study, the total cholesterol and the LDL cholesterol was taken as a marker of who recovers from severe infections. So this group took 179 patients. They had to exclude a number of patients for different reasons. These patients were all critically ill in an intensive care unit called the ICU. And they analyzed the people who survived the infection, which is out here, 65, and people who did not survive the infection, which is 52, to see what was different about their cholesterol profiles. And what was found, and out here in the dark circles are people who lived through the infection, they survived. The open circles are ones who perished. And here is total cholesterol. What we found is that the total cholesterol was higher in the group that survived compared to the group that did not survive. that the LDL, the so-called bad cholesterol, was higher in the group that survived compared to the group that did not survive, that the triglycerides were higher in the group that survived, and that the HDL, the cholesterol that clears fat from circulation, was high. By the way, I'm trying my best not to use the term good and bad as far as LDL cholesterol is concerned and HDL cholesterol is concerned, because I think these labels require changing. So what does LDL do when it is not modulating inflammation or doing its work in terms of host defense and protecting us from invaders like bacteria and viruses? So the LDL is a molecule through which we recycle cholesterol. So the liver picks up this LDL molecule through the Ldl receptor so that the liver can do a number of things. It can make bile so that we can absorb fatty acids and fat, as well as absorb fat-soluble vitamins. It also eliminates cholesterol and bile based on our needs. So the body has a fine homeostatic mechanism of how to regulate the amount of cholesterol we have. The vascular smooth muscle cell that lines our blood vessel wall and the endothelial cells, which are part of the lining, which is the area which gets exposed to blood flow, they also have LDL receptors, and they pick it up for cell repair and for other uses inside the cell. So cholesterol is an essential component of every cell, without which our body will not be able to function. And our body has mechanisms to pick up this cholesterol, because the liver is assigned with making about 70% of our body's cholesterol. So it makes the cholesterol for cell repair. And one good way to illustrate that cholesterol is needed for growth is by talking about a newborn. A newborn has one of the highest production rates of cholesterol and the lowest amount of LDL cholesterol. And the reason for that is that the newborn is in a growth spurt. It's making new cells, new tissues, and those require cholesterol. And that is supplied by the LDL molecule. So in this depiction, I have a cell membrane. A cell membrane is the covering of a cell. Every cell, it protects itself, and the cell membrane gives it the fluidity and the flexibility to the outer covering of a cell. And an important component of that is cholesterol. So I'm going to see if this depiction plays. And what here is shown, a cell membrane with a phospholipid bilayer. There are cholesterol molecules in that. As the cell membrane gets damaged, the cholesterol is removed, but then here comes the LDL. It's supplying the cholesterol so that the cell membrane can be repaired. So what this cartoon showed is damage to the cell membrane, the cholesterol being removed, and then subsequently repair of cholesterol through the LDL molecule. Does this really exist? So there are clinical studies, basic science studies, that show that here is an LDL molecule. It's getting into the cell through endocytosis, receptor-mediated endocytosis. That is the LDL receptor taking it in. It goes through a series of chemical reactions. Free cholesterol is then released, and this cholesterol then goes to an area where the cell membrane is damaged so that there can be repair. Now this is also shown in here in this study that was done with cells. And in this group, what they did was they removed LDL from the medium in one group, had LDL present in another medium, and they tried to find out whether these cells regenerated. So here is the leading edge of regeneration in here. This is cells that don't have LDL in here. And the regeneration is quite slowed down. In other words, the leading edge of new cells is significantly reduced. Whereas in this case, in which the LDL is present, the leading edge of repair, you see more cells that are present and repair is happening. Now, is this relevant clinically? When you have a blood vessel, the blood vessel gets damaged because of high blood pressure, because of infection, inflammation, the lining of the blood vessel wall gets damaged. And when the lining gets broken up, it can create a blood clot. And as these blood clot grows, it can block the blood vessel and cause a heart attack. So that is the break in the blood vessel wall, blood vessel lining damage or endothelial damage. And if left unchecked, a blood clot grows and blocks the blood vessel, causes a heart attack. In this situation, there is repair of the lining. Perhaps LDL is involved in it, and that's why it is such an important molecule for us. And LDL is repairing the damage and preventing a stimulus for blood clot to form. So when viewed from this paradigm, the LDL is not bad and it's helping repair injury. Now I want to get into the part as to why I think oxidized LDL is maligned for the wrong reason. And in order for me to explain that, I have to tell you what PCSK9 does. So PCSK9 is a molecule that our liver makes, and it's depicted out here. As the PCSK9 increases in the setting of an infection or inflammation, it's an acute phase reactant, which means that in the setting of infection or inflammation, the liver makes more of this. This PCSK9 comes in and it removes the LDL receptor from the liver. So the LDL receptor is the one that is sucking up LDL from circulation, the so-called bad cholesterol. And when you remove or downregulate the LDL receptor, the LDL is not removed from circulation. A lot more LDL molecules will then be found in the bloodstream. And when a lot more LDL molecules are found in the bloodstream, LDL has many biologic functions that I have shown here on the right of that slide. And today we're going to focus on host defense in terms of neutralizing lipopolysaccharide. And we have already talked about cell repair, the few other things like cancer surveillance and energy delivery have been expounded by other people. So we are in the habit of using drugs to reduce LDL, and one of them is this powerful medicine called PCSK9 inhibitors, and they have been used extensively. In my previous presentation, I talked about they're about $5,000 a year to use this. And what PCSK9 inhibitor does is that it comes and neutralizes the PCSK9 that the liver is making in the setting of an infection or inflammation. And when PCSK9 is neutralized, the LDL receptors are not removed. These are LDL receptors on the surface of the liver. And as the LDL receptors grow in number, they soak up the LDL. You can see the LDL is kind of being removed from circulation, and very few molecules of LDL are left in the circulation. So your LDL level drops dramatically to as low as 30 milligrams. I have seen people using PCSK9 inhibitors who have an LDL value close to 0. And I have shown in the slide that as you reduce the LDL to 0, on the right side of the slide, some of the biologic functions of LDL, I have made them disappear. PCSK9 is an evolutionary conserved molecule. It has many important functions. And one of the important functions is to modulate host defense, to modulate inflammation, to provide energy. Can we illustrate that? Are there studies that help us understand this better? So here is a paper that is describing how our body deals with LDL and how it uses it. On the left panel is a normal situation in which there is no infection and inflammation. Here is the LDL molecule shown in yellow. This is the LDL receptor on the liver cell that is taking up the LDL into the liver cell, and it's removing the LDL as bile. The bile is then being used to absorb fat that we eat, to absorb cholesterol, to absorb fat-soluble vitamins. And the amount of bile that is made is not just for that, but also for elimination of cholesterol. You can also see that the endothelial cell, which lines the blood vessel wall, is taking up some cholesterol for its own functions. And the amount of cholesterol that is being diverted to macrophages, these are cells that fight infections, is very low because the expression of scavenger receptors, and we'll get into that in a little bit, is not very high. Now come to the right side of the slide and you see this is a situation in which there is infection and inflammation going on, and the body is very smart. Evolution has designed that when there is infection and inflammation, that the levels of PCSK9 go up, and as the levels of PCSK9 go up, it down-regulates the LDL receptor. So the liver cell is not picking up the LDL. The reason it's not picking it up is twofold. One is that it wants to leave the LDL. It's one of our trenchmen. It's trying to fight an infection. It's trying to fight inflammation. So it wants to leave LDL in the circulation so that it can fight infection and inflammation. And out here is shown a modified LDL particle. This LDL particle either has lipopolysaccharide or viral particles. It's gone through oxidative damage, and you don't want to pick up damaged LDL into the liver cell because that needs to be cleared. This is an injured fireman that is cleared by upregulating or increasing the amount of scavenger receptors on macrophages, so that healthy cells like the liver cell and the endothelial cells don't take something that has been damaged. This is a mechanism that our body has designed. And to illustrate this a little bit further, the damaged LDL particle is taken up by scavenger receptors out here. There is also non-receptor mediated endocytosis. In other words, the macrophage is capable of picking it up without a receptor. The macrophage increases the expression of something called toll-like receptors, or TLR, which is shown out here in this Y kind of cartoon. That also picks up bacterial and viral products and picks up oxidized and damaged LDL. And when the macrophages picks up a lot of this, it can become fatty and it's called a foam cell. But what's also happening out here is that the PCSK9 is preventing the uptake of the oxidized LDL by normal cells like vascular smooth muscle cells and by endothelial cells by down-regulating the LDL receptor. Now, it's only in the setting in which we let inflammation continue without it being abated. In other words, the macrophages are there to help deal with injury and clear injury. But if you're eating a persistently processed, carbohydrate-rich diet in which your insulin levels are persistently high, causing insulin resistance, and I have gone into what insulin resistance is before in different presentations. Insulin resistance leads to persistent inflammation. Inflammation does not go away, and when that doesn't go away, the macrophages perhaps cannot have the capacity, they get overwhelmed, and that leads to the fatty streaks and the plaque. It is not right to implicate oxidized LDL in that process. The root cause of that is not oxidized LDL. Oxidized LDL is something that has gone in there to protect the inflammation. It's not a cause of that inflammation. Getting rid of LDL is not going to take care of that problem. But getting rid of inflammation by root causes, what is causing the inflammation in the first place, is the method of dealing with it. Now, is there evidence that when there is a high PCSK9 level as a result of inflammation and infections, whether the scavenger receptors are upregulated? And in this study, it is showing you that PCSK9 levels are going up in the setting of an inflammation and infection. And as the PCSK9 levels are going up, the scavenger receptors, this is the LOX1, the oxidized LDL receptor, the scavenger receptor A, the CD36, these are all in the family of scavenger receptors. In this Western blot, which is a measure of the amount of protein of these receptors, you can see that the expression of that protein keeps increasing as the PCSK9 levels go up. And shown here on the right side, as the PCSK9 levels are going up out here, you can see PCSK9 levels going up, the LDL receptor is going down so that it's protecting the liver and other cells and preventing them from taking up oxidized LDL and leaving LDL there in the circulation to do its job. So, is there other reasons why the PCSK9 downregulates the LDL receptor? So in this slide is shown an LDL receptor, and by the way, there's a little mistake in this slide. Even though this is an LDLR, what I wanted to show here was this is a family of LDL receptors. This one should have been called the LRP. So LDL receptors is an LDL receptor family that is designed to deal with lipoproteins. It's designed to clear fat after we eat fat, which is the function of the ApoE, the VLDLR, and the LRP receptors. And the number of these receptors is regulated by PCSK9. As the PCSK9 levels go up, these receptors go down. It's important to point out that these receptors are present in a lipid-rich domain of the cell membrane that is cholesterol-rich, as shown here in yellow. These receptors are also present in something called caveolae, which are invaginations of the membrane. And in the invaginations of the membrane, which are cholesterol-rich, these receptors are positioned. So not only am I showing the LDLR family of receptors, but I'm also showing the ACE2 receptor. The ACE2 receptor is through which the SARS Co V2 or the novel coronavirus gains entry. And the reason I point this out is because there is a hypothesis that as PCSK9 is removing the LDL receptor from the lipid rafts, any other receptor that's present in the lipid raft would also be removed. So it's quite possible that PCSK9 is designed to protect us from viral infections. We know that that is true for the hepatitis C virus. And by the way, there are several infectious agents that try to co-opt the lipoprotein and the lipoprotein receptor system in order to gain access to our cells and cause infection. So it's possible that many of these bacteria are designed to use our cholesterol system to gain infection, but it is importantly a method by which we fight infections. So I'm taking this opportunity to demonstrate that there is a possibility that as you downregulate the LDL receptor family, and take it up in the lysosome, which is the garbage disposal mechanism of the cells, and you're breaking down the receptors. If there are other receptors in the lipid domain that come into the lysosome, such as the ACE2 receptor, it can potentially prevent viral infections. And in this day and age where the novel coronavirus is important, this is the mechanism that we should consider and study so that we understand the human body better. Now, here I am using a cartoon, and this is not a perfect cartoon. There are some mistakes in it. But here it's pointing out the LDL receptor family, the LDLR, the VLDLR, the ApoE, the LRP, and they are being downregulated by PCSK9. And as these are being downregulated, the elaboration of the scavenger receptor increases, the LOX1, the scavenger receptor A, or the SCA. And I have made a few errors here. I wanted to show you that this is a macrophage that is increasing the scavenger receptors, but basically it illustrates my point. And my point is that oxidized LDL, instead of being picked up by the LDLR, should now be picked up by the LOX receptor, by the scavenger receptors, so that the macrophages can deal with damaged and oxidized LDL. So I wanted to remind people that there is ample evidence that PCSK9 downregulates the LDL group of receptors, upregulates the scavenger receptors so that it can deal with oxidized and damaged LDL. And the reason it's doing that is that LDL levels increase and is reserved for dealing with infections and inflammations. That oxidized LDL is not picked up by healthy cells because oxidized LDL is damaged. And that monocyte and macrophages, the cells with the greatest ability of removing infections and inflammation, are now removing these first defenders, which is the damaged LDL particle, and trying to convey that through this slide. Now, PCSK9, in order to be a little bit more complete, not only downregulates LDL receptors, not only upregulates scavenger receptors, but it alters the genetics of the cells so that there is more cholesterol efflux. By that I mean that more cholesterol is released into the bloodstream through different mechanisms. One of them is the ABC cassette, which is expressed through turning on some genes so that more cholesterol is available to fight infections. There is also transcriptional, which means genetically reprogramming the cells so that they are releasing more of the scavenger type of receptors and other functions. So PCSK9 is an important molecule that our body is elaborating. And to just inhibit it without thinking about the many beneficial aspects of it and without completely understanding it, I think by mainstream medicine could be fundamentally wrong. So I'm asking these questions. Is it possible that we are ignoring the evolutionary design of our body, of making PCSK9, of making LDL available, of the LDL receptors in a coordinated function so that they modulate the LDL's ability to defend against oxidative injury, so that LDL as an innate defense mechanism is utilized properly, that LDL is used for cell repair, that LDL, which is important to supply raw materials and energy, is present in the setting of infection and inflammation. I also want to point out that oxidized LDL is perhaps a primary defender a primary defense mechanism to soak up oxidative injury, that it is not a cause of oxidative injury, and removing LDL, which is a firefighter, is not the method in which we should deal with inflammation. That inflammation is the root cause of coronary artery disease, of blockages, and that the PCSK9 mediated downregulation of the LDL receptor is a mechanism to protect the host, protect the normal cells from a molecule that has been damaged in the process of protecting the body, and I'm referring to the oxidized LDL. So in closing, I want to give the view and the paradigm to my viewers that if you make proper interpretations based on science, based on common sense, based on data, that oxidized LDL is an injured firefighter and that it is not an arsonist that is the primary cause of vascular disease. Thank you.